we're going to get into kind of discussing electromagnetic regulation of the cell. You could say this is a new frontier, although in saying that, we'd really have to recognize the contributors that have laid the groundwork, because in a very fundamental way, the groundwork has already been laid. I'm talking about the proprietors, or we could say the principal architects of this new field that we're going to call the field of radiobiology. These would include Nikola Tesla, George Lakhofsky, Royal Raymond Rife. I'm just going to stick with those three for now. These are the principal founders, really, of the field of radiobiology. These are men with the utmost integrity. Shall we say they probably all possess a level of scientific genius that is unparalleled. For more than 22, 23 years now, I've really fixated my limited intellect in comparison on understanding their work, understanding its application to human health. So let me touch briefly on the significance of these men, because I think it's very important. We can't assume that everybody on here has a really good understanding of the principal architects, the pioneers in this field of radiobiology. But when we think of Nikola Tesla, we think, here's arguably the greatest inventor that the world's ever seen, though the powers that be have obviously attempted to expunge much of his work from the records. And I think that's been really, really hard to do, particularly after the rise of the Internet. Think about it. Here's the guy. He's the founder of robotics, computers, missile science. If you look at his original work, it was so transforming that it was virtually without theoretical precedent. This is the guy that that paved the way, really, for things like satellites, microwaves, beam weapons, cellular communications, and nuclear fusion. And the ironic part of Tesla is that he really remains one of the least known scientific pioneers in history, and yet here's the guy that you should think of every time you go to the wall and plug anything into the wall. You should be thinking of Nikola Tesla. He was the inventor of alternating current, or what was at one time called the rotating magnetic field. Everything that you plug into the wall, that's alternating current, and this is your guy. Okay, among the many amazing inventions that were attributed to this scientific genius, Tesla, was the Tesla coil, which is simply a high-frequency oscillating coil with primary and secondary windings. We may get into discussing that a little bit. We use it extensively in many of the technologies that we've developed. An entire branch of medicine was established on the basis of the Tesla coil. And that's really important to understand. Historically, in fact, at one point, various Tesla-based electrotherapeutic devices were available in the Sears catalog. I kid you not. So the success of these instruments ultimately led to the suppression of the devices, anything really within bioelectrics or radiobiology, by the medical establishment, which I'm sure everybody is just absolutely shocked, right? Suppression by the mainstream, it's unheard of. So over a period of roughly 8 years, Tesla developed about 50 types of oscillating coils. I mean, if you come into our lab, you're going to see cylindrical coils, cone coils, flat coils, spiral coils, every kind of coil imaginable. But the most amazing Tesla coil for electrotherapeutics that he developed was what we call a magnifying transmitter. So imagine a coil that's connected through wireless induction of the secondary winding of the Tesla coil. So you have your Tesla coil as the primary winding, the secondary winding that's kind of inside, and then that secondary is connected with a magnifying coil that is connected wirelessly. So it's just connected by induction. That was really kind of the big breakthrough in resonant therapy. And that was Nikola Tesla. A lot of people, when we think about George Lakhofsky, they don't realize that the vast majority of George Lakhofsky's work with the multi-wave oscillator came from Nikola Tesla, which really brings us to George Lakhofsky, a French engineer, Russian-French engineer who actually collaborated with Tesla. He was having a difficult time with his original unit in the 20s, and actually, as I remember, he flew to France, met with Tesla, and so a lot of the multi-wave oscillator, we don't know how much of it was actually developed by Tesla, but we do know that Lakhofsky consulted with Tesla in expanding on the therapeutic benefits of high-frequency oscillations. So Lakhofsky went on to develop what we call the multi-wave oscillator. The multi-wave oscillator was in kind of a major improvement in Tesla's magnifying transmitter, largely because of a more robust theoretical framework, which stated, and this comes from Lakhofsky, which stated that diseased cells are cells that vibrate out of harmony with the rest of the body and with lower electrical charges, which is extremely interesting if you consider that it was in the early 20s that this all came about, which was, I think it was probably a decade later that you had the Nobel Prize winner Otto Warburg confirming that healthy cells of a newborn infant, let's say, have cell voltages of minus 70 to minus 90 millivolts. That was a decade after Lakhofsky. Otto Warburg came and published those findings. As we age, as we get older, the theory from Lakhofsky is that the electrical charge around the cell decreases, and it can decrease to as little as 35 millivolts by age 70 or so. But if it's a disease cell, let's say it's a malignant cell line, they have membranes with excessive positive charges around 50 to 60 millivolts on the plus side, plus 50 to 60 millivolts. I've seen them as high as 70 millivolts. This is because in a cancer cell, that membrane will kind of, all those protons will leak, and we'll get proton accumulation, and those are all positively charged. So we get plus 70, plus 50, 60, 70 on the millivolt potential in a diseased cell. So the key to Lakhofsky's success was developing an instrument, and he knew he had to really develop one that could broadcast not just one frequency, and that's rife, one frequency. We'll talk about that in a little bit, but frequencies over a very, very broad range from around 700,000 cycles per second to around 300 trillion. And he had this crazy idea that the primary nuclear structure of the cell consisted of a double helix. Okay, this was back in the 20s that he had the idea that the nuclear structure of the cell was a double helix antenna capable of being stimulated by external frequencies. So this is the guy, that postulated the existence of the double helix. What was that? 30, 40, 50, so about 30 years before Watson and Crick discovered the chemical structure of DNA. 30 years prior to that. Here you have Lakhofsky postulating the existence of the double helix. So Lakhofsky believed that the cell had to be understood basically as an electronic circuit. It had resistance, it had capacitance, it had inductance. It was an electronic circuit. Three electrical properties that, when properly configured, with the correct external frequencies, could bring the cell back into a state of equilibrium or back into a state of homeostasis. And let me quote from Lakhofsky here. Quote, this filament, which may be the RNA-DNA complex, is always in the form of a spiral or helix. In other words, a coil. Therefore, each will react as a tuned circuit if its resonant frequency can be approximated by an external oscillating coil. I postulate that by exciting the nuclei with electromagnetic energy, a charge can be induced by the long-established principle of electromagnetic induction. This demonstratively raises the energy level and perhaps the vitality of every cell in the field simultaneously. Since each cell is an individual and of slightly different physical dimensions, the exciting wavelengths must be multiple and must span a broad frequency spectrum. End quote. So, and that's exactly what Lakhofsky was able to accomplish with this special antenna, this multi-wave oscillator antenna that he configured based on what is referred to as the golden ratio. I don't know if you guys are familiar with the golden ratio. I won't delve into that too much here. Maybe we'll have a lecture just on the golden ratio, but suffice it to say that Tesla, Lakhofsky, many others recognized that there are patterns that naturally occur in the universe, patterns that we've discovered in life, in galaxies, in star formations, in almost all the natural systems. Some of these patterns are the golden ratio. Some of them relate to what we call sacred geometry, and perhaps we'll do a lecture on that subject at some point on its own. But the antenna worked on the principle of induction. Let's just stick with that. With the inner, so it had 12 rings. The inner 11 rings of the antenna were connected only through secondary induction. It was only the very first outer ring that was connected to the secondary of the Tesla coil. All other 11 rings just kind of free-floated. And if you've been to our center, you've seen this ring configuration. They're not touching the primary ring that is energized by the output of the Tesla coil secondary. But this inductive configuration maximizes the natural spread of the frequencies over the broad range. And that's what you've got to have, is you have to have a broad range from about 700,000 cycles to 300 trillion cycles per second, which allowed every cell and every structure within the cell, particularly the helix filament, to resonate at its natural vibratory rate. Again, generating this really, really broad spectrum. So this enabled the multi-wave oscillator to ensure what Lakhofsky would often refer to as dynamic equilibrium of the cells by creating a multiplicity of sideband harmonics. The subject, will take the person, set them in a chair, tell them don't touch the coils, because they'll be in for a 300-volt discharge. Of course, it's not lethal because it doesn't have a lot of amperage in it, but it can scare you. Well, it's like something out of Frankenstein's lab. But you put them between the coils, and essentially, when the device is turned on, the field can be readily observed just by placing like a light bulb in the field. Like we take a fluorescent light and we'll just wave it through the field, and you'll see that thing light up through induction. And so they sit in that field, and because they're in the field, they actually become part of the Tesla coil circuit through what we call capacitive loading. So they're part of the circuit now because they're resting in the field. Lakhofsky believed that disease, if he was going to define disease, he'd define it as a battle between the oscillations of the organism and the oscillations of the cell. When the organism gained the upper hand and oscillatory disequilibrium of the cells resulted, if the proper frequencies were introduced and kind of upregulated those structures, then the cell became the predominant force and health restoration was the result. So if we look into, I want to get into some interesting things here with you that I found in the Anti-Aging Medicine Journal. But if you use, let me just read it here. If you use electromagnetic frequency, which is matching the spinning frequencies of hydrogens of a particular aging tissue, its atoms and electrons will be collectively excited by resonance. So the electrons will emit their photons together and will produce an enormous bundle of coherent laser light in the cells. And of course, in one of my later lectures, I'm going to get into the works of Potts, and we're going to discuss biophotons. The article goes on to say, since these photons can circle between molecules without loss of energy over a long time, for hours, their coordinated actions exert the astounding order and repair effect. So that's Lakhofsky. Multi-wave oscillator. In Delta has spent, my goodness, we've spent a lot of years reverse engineering and developing different multi-wave oscillation devices. GenPulse, T-Flux oscillator, Tesla, radiocellular oscillators, full-blown large magnetic oscillator devices, magnetic oscillator tables, magnetic oscillator beds. We've got some cool stuff that we're developing right now. And one of the first devices that you'll have access to, actually, and we'll probably initially only give our coaches access to this, will be the T-Flux oscillator, which I'm not going to get into here because we need really it needs a lecture of its own. But if you want to learn a little bit about that, you can go watch something on like Tesla Cellular radio oscillator, but that only gets into one specific mechanism of cytochrome C, we'll get much broader as to what the oscillator does when we get into that video. But you'll see that become available soon. In fact, it's the store is being worked on right now where we'll be able to give our coaches access to that. So let me review a few facts about Lakhofsky, just kind of in summary, because I want to make sure you guys understand the significance of Lakhofsky. He was a radiobiologist, or what we'd call a biological physicist, was credited as being the first to predict the existence of the double helix. He postulated that the living cell essentially possesses attributes which are normally associated with electronic circuits. He found that the organisms typically responsible for compromised health states produce different frequencies than your normal healthy cell, and that by increasing the amplitude of the oscillations, I'm just repeating here, the oscillations of the cells, if you increase the amplitude, that kind of overwhelms, kind of dampens out the oscillations produced by the organisms. So the organism is then either converts to a non-pathogenic state, or the organism actually dies. There are a couple different mechanisms there that happen. On the other hand, if he pumped up the amplitude, and he did this, he experimented with taking cultures, microorganisms, then he'd pump up their oscillations, and then they would gain the upper hand and cause the health of the individual or the cell culture to decline. And the way that he was looking at it was, hey, if this is really a battle between the resonant oscillations of the host cell versus the oscillations that were emanating from these pathogenic species. We also know that historically, Lakhofsky's success, or the word of Lakhofsky's success with his multi-wave oscillator, we don't hear about him now, but if you went back to the 20s and 30s and 40s, you'd find that news quickly spread throughout Europe. In fact, he was widely celebrated in Europe. And you could find significant reference to the magnitude of his work in most of the medical journals of his time that were circulating, at least through Europe, not in the States. But in fact, the Irish Journal of Medical Science, which was a kind of a review, or did a review of Lakhofsky's work, and I'm going to read from that journal. I want to say that was 1929, I believe. It says that Lakhofsky's work is now being given serious consideration in all countries. His general theory is one which physicists, biologists, and doctors can no longer ignore. Dr. Postma, a very renowned researcher, wrote the book Life in the Waves, which is a book that right now we're in the process of getting translated from French. We have the book. We've located a French copy. It never was translated into English, but that'll also be available. I think it'll be the first English translation of Life in the Waves by Dr. Postma. Will be available in our library, which, by the way, we are getting ready to translate some other books as well, and we'll keep you up to speed on those. Some of them are in German, and we just don't have the English counterparts yet. But Dr. Posta proved mathematically, and the journal entry goes on to say that Dr. Posta proved mathematically and experimentally the accuracy of Lakhofsky's theories. That's stated in his book. He kind of outlines that. And I quote from his book here, said that the work of Lakhofsky surpasses that of Pasteur and Edison in its scope. I want you to think about how potent that statement is. The work of Lakhofsky surpasses that of Pasteur and Edison in its scope. That's pretty powerful statement. European historians estimate that approximately 35,000 practitioners were actually using Lakhofsky's technology in and around Europe between 1931 and 1953, and yet he's been essentially, again, expunged from the records. Seems to be a trend here. That kind of brings us to Royal Rife. I want to kind of throw him into the mix here, another important contributor to the field of radiobiology, curing cancer, curing several other types of diseases, and his research was destroyed. Again, surprise, surprise. His associates were obviously harassed and killed, and they burnt down labs. But Reif was arguably one of the most brilliant scientists ever to walk the planet. He had 14 awards from government for his research. He was given an honorary doctorate, or I say an honorary medical degree from the University of Heidelberg in the midst of World War II in Nazi Germany. So most Americans don't get an honorary doctorate from a University of Heidelberg right during Nazi Germany. The thing about Reif, though, even more impressive, is if the technology didn't exist, he'd just invent it. He would just machine it. He was financed by millionaires like Henry Timken of Timken Bearings, so he basically had a blank check. He had the liberty of directing some of those resources into constructing a highly sophisticated lab, probably the most sophisticated lab of its kind back then, which was in the basement of Timken's garage, or guest house, I should say. He went on to invent what was called the universal microscope, which most of you know about, which was featured alongside the electron microscope by the Smithsonian Institute in Europe. This thing had some 5,600 moving parts. It was capable of seeing matter that was smaller than a wave of light, which even today they say is impossible. But Reif was he was the first one to really see a live virus. Even today with the electron microscope, you can see the virus, but what you're looking at is dead because it's completely bombarded with electrons and there's just nothing going on there. But with Reif, he could actually view the bacteria or the virus as a live organism. He was able to determine that each of the organisms, similar to what Lakhofsky identified, emitted a distinct pattern of light. Of course, he was able to kind of visually confirm photon emissions. So what Reif did is he started experimenting with instruments that he invented that oscillated at the frequencies that he determined from the organisms that he had under the microscope. And what he discovered is that by playing back their own pattern of oscillation and then just slightly modifying it, he could destroy the organism without affecting any of the healthy tissue around it. So he could kill a particular virus, bacterium, using light rays, using radio frequency-assisted catalysis, really, light rays that were absolutely harmless to the host animal, but very deadly to the microbe. Ultimately, you know the history. Reif was, able to isolate the virus or the filterable bacteria that causes cancer. He proved out Koch's postulate. There were actually two forms, of the what he called a virus. The only reason he called it a virus, incidentally, was because it was less than 1 micron. It passed a 1 micron filter, but the BX was not a virus. It was a bacterium. He made that clear if you go back into the notes. But the definition of a virus was anything that passed a 1 micron filter. But he named he had two variants, the BX. Now you know where the word BX comes from, and the other was the BY. But Reif ended up, he hooked up with Arthur Kendall of Northwestern University, which was probably one of the best bacteriologists in the country at that time, and they perfected a means of culturing filterable forms of bacteria. He was able to selectively destroy the organism with radio frequency and spent the next several years kind of tweaking that technology, perfecting it, and by this time he was, kind of a rock star. He was really on at one point he was on every newspaper in the country. They had held that honorary banquet in his honor and to celebrate the end of all disease, and I'm sure many of you know the history there. But Reif eventually went public in the University of Southern California, as you remember, sponsored a special medical research team that conducted initial clinical trials. I believe that was at the Scripps branch, if I remember right, and the results. 16 out of 16 terminally ill cancer patients participated in that trial, were cured of cancer. And I believe all but 2 were cured in the first 6 weeks, and then eventually over the 130 days, 16 out of 16 were pronounced cured. And the rest of the history here. Let me fast forward 85 years into the future. Let me read you an article summarizing some very interesting findings. The title of this article is is human cells have electric fields as powerful as lightning bolts. Using newly developed voltage-sensitive nanoparticles, I'm reading this article here, researchers have found that the previously unknown electric fields, think about the words here, previously unknown electric fields, hopefully some of you will chuckle, inside of the cells are as strong or stronger as those produced in lightning bolts. Previously, it has only been possible to measure electric fields across cell membranes, not within the main bulk of the cell. So scientists didn't even know cells had an internal electric field. More chuckles, right? This discovery, because this is modern, this is the University of Michigan, this discovery is a surprising twist, the article goes on to say, surprising twist for cell researchers. Scientists don't know what causes these incredibly strong fields or why they're even there, but now, using new nano tools, such as voltage-sensitive dyes, they can start to measure them at last. Researchers believe they may be able to learn more about disease states, such as cancer, by studying these minute but powerful electronic fields. University of Michigan researchers, led by chemistry professor Raoul Copeland, encapsulated voltage-sensitive dyes in polymer spheres, just 30 nanometers in diameter, testing these nanoparticles in the internal fluid of brain cancer cells. Copeland found electric fields as strong as 15 million volts per meter. Okay, it's up to about 5 times stronger than the field that's found in a lightning bolt. 15 million volts per meter. However, this discovery goes beyond being incredibly interesting, the article states. The findings will likely change the way researchers look at disease. Really, okay. It goes on to say they've developed a tool that allows you to look at cellular changes on a very local level, said Piotr Grodzinski, director of the National Cancer Institute Alliance for Nanotechnology. In Cancer in Technology Review, Grodzinski believes many developments in cancer research, for example, over the past few years, have been reactive rather than proactive, despite how far cancer treatments have come. The way that cancer and other diseases progresses at the cellular level in the first place is still not well understood. With a better understanding, researchers could improve diagnostics and care. You notice they say nothing about treatment, really, because everything's about diagnostics and care and longevity of treatment, right? It goes on to say this development represents an attempt to start using nanoscale tools to understand how disease develops, says Gudzinski. Copeland has developed encapsulated voltage-sensitive dyes that aren't hydrophobic and can operate anywhere in the cell rather than just in the membranes. Because it's possible to place his encapsulated dyes inside of a cell with a greater degree of control, Copeland likens them to voltmeters, nanovoltmeters. Do not perturb the cellular environment, and you can control where you put them, he says. The existence of strong electric fields across cellular membranes is accepted as a basic fact of cell biology. The fact that cells have internal electric fields as well, however, is a whole new revelation. It says scientists previously did not know of the existence of internal cellular energy fields. Let me read that again just so you guys can get a laugh out of this. Scientists previously did not know of the existence of internal cellular energy fields and are just in the earliest stages of understanding the phenomenon. Copeland presented his results at the annual meeting of the American Society for Cell Biology this month. There's been no skepticism as to the measurements, says Copeland, but we don't have an interpretation. Daniel Chu of the University of Washington in Seattle agrees that Copeland's work provides proof of concept that cells have internal electric fields. And then he goes on to say it's bound to be important but nobody's looked at it yet. So I really had to chuckle at that because we get two things out of that. We don't have an interpretation, and nobody's looked at it yet. And it's like our inability to learn from history is a very complex set of problems, folks. As I've mentioned before, these examiners, you get somebody that's doing their research. Let's say somebody's working on their doctorate. They just have this ossifying view, the thesis examiners, I should say, if somebody's going to get a thesis approved, have this view that, hey, anything over 10 years old, forget it, we're not going to look at it. I've given this example before. You're a physicist, you're wanting to do your thesis, you're a doctoral student wanting to do your thesis on overcoming the Fraunhofer diffraction limitation in optics, like Royal Raymond Rife actually did, right, in building the universal microscope. And you have to, you have no recent research to support that research topic. It's very unlikely that the topic's going to gain approval. Your thesis examiner is going to look at you like a deer in headlights. So in general, there's this absolute and utter lack of respect for non-replicated vintage research, which has, in my opinion, carried a very heavy cost. And unfortunately, researchers, choose says it's bound to be important, but nobody's looked at it. This is the first time we've ever seen it. My goodness, this stuff was being postulated, long before, right? As the era of radiobiology dawns, okay, this is the second dawning of this era, I think in some respects it's probably easier to ask when we think about, the bioelectric nature of ourselves, what aspects of physiology do not pertain to the bioelectric theory? That's probably a more appropriate question to ask. Which aspects of physiology don't pertain to the bioelectric theory of health? Is the body truly electric? I would submit the answer is yes. Think about we're often talking about BX mechanisms, cellular metabolism, and cancer as a metabolic disease. And you think about the metabolic machinery of the cell, and this is very interesting when we think about how oxidative processes, and particularly oxidative phosphorylation, relates to energy. We eat food, it ends up as pyruvic acid is broke down, the electrons flow, they generate ATP, right, for the cell. The cell's process of oxidizing glucose and fatty acids, here's the question, what happens when that process is impaired? Some of the first things that you'll notice, you look at a sick cell like a cancer cell, what happens the minute that it starts moving away from the oxidative processes, moving away from oxidative phosphorylation, moving toward fermentation? What happens? Well, one of the first things that you notice is the membrane potential, the electrical potential in that membrane begins to immediately degrade, both in the cell wall and in the mitochondrial membrane, moving from that optimal negative 70 millivolt potential all the way down to 30, 40, 50, I think I've seen as much as 70 millivolts on the plus side, where a disease cell starts to depolarize. So as oxygen utilization declines, the bioelectric status of the membrane immediately follows, which brings us to the obvious question, is oxygen electric? Well, of course, the answer is, I wish we had a survey on here. Survey says, yes, oxygen produces radiant energy, right? And radiant energy generates electric currents across all of the stinking biological terrains that we know. But we have a lot to consider. It's not just as simple as an electric field, and I think the conventional realm is still mostly focused, and they're more than happy to accept potential energy in the form of chemical bonds. That's chemical energy, right? Because that fits better in with the development of pharmacological agents. And admittedly, the conventional realm readily accepts the electrical properties of cellular constituents. But there is certainly more to consider than chemical and electrical energy. There are some incredibly relevant areas in chemical and electrical, don't get me wrong, but we really can't ignore the role of three particular forms of energy, and I'm not going to get into this week because we're just not going to have enough time, But these are probably, at least to me, the most interesting, and they are namely biophotons, radiant energy, and the quantum field. These are three areas, and I suspect that each one of those will probably need its own lecture. I think I'm going to leave that, we'll leave it at that for now. I think we'll get into biophotons. I don't know, maybe we have a combined lecture. Maybe we discuss them separately and break that into three lecture topics